Hi, everyone. Uh, let's get started. We have a lot to do today. I think we're going to start with the genetics questions and the biotech questions. Those are the harder topics, right? And we might have to go a little bit over time, to finish up everything. This thing is going to be recorded, of course, and be uploaded to eCentennial afterwards. So let's get to it. First question, explain the significance of crossing over. How, why, where, when does it happen? Okay, so crossing over, the purpose of crossing over is to create genetic variation, okay? And that happens during meiosis, okay? Specifically, it happens during the prophase 1 of meiosis. So let's write that down real quickly. Um, how? Okay, so how does it happen? Well, you remember, right? We have the homologous chromosome coming together. Okay, so the homologous chromosome is going to come together to form tetrad. Okay, so this is called a tetrad. And these are locations of crossing over, right? Crossing over, which is the exchange of genetic information between homologous chromosomes. Okay, this generates genetic variation. And when does it occur? It happens during prophase 1 of meiosis. Okay. Next, what is the purpose of meiosis? Where does it happen? What is the result? The purpose of meiosis is to create gametes, right? Which is needed for sexual reproduction. Where does it happen? In the gonads, right? That's like testicles and ovaries. And what are the results? 4 genetically different haploid cells. Okay. It's pretty basic stuff. Next, what is non-disjunction? When does it occur? What is the consequences? Non-disjunction is basically the failure for chromosomes to separate during metaphase, during anaphase 1 or anaphase 2. Okay. And what okay, so that's the what and the when. What is the consequence? Consequence of this is you will end up having some gametes will have extra or missing chromosome. Okay, depending on whether it is on the receiving end of that chromosome that didn't separate, or if it was missing that particular one. That's all you have to know for non-disjunction. Okay. Let's see. Practice all the different types of genetic problem: mono, hybrid, dihybrid. Yeah. So there is going to be some of those questions again, like the Punnett square ones. You will see them maybe in multiple choice. You will also see them in the long answer, like written response section as well. So make sure you redo some of those word problems from the study guide. And I know I didn't hand back quiz number 4, but quiz number 4 is essentially same as the review sheet that I gave you, right? So if you were able to do those problems on the review sheet for quiz 4, you should be okay. Let's move on to the next one here. Now we did this question a while back. It's one of those like poll questions that I do in class with you in the PowerPoints. So let's just go through it again. How many unique gametes could be produced through independent assortment by an individual with this particular genotype? Independent assortment just means that how you sort one gene into the gametes is an independent event of the next gene. So whether you get the big A or small a in one gene is not going to affect whether you get a big A or a small b in the next gene. So let's take a look here, because we have 2 choices for A, right? So we're going to put in number 2, right? You can get big A or small a. You have 2 choices for B, 1 choice for C only. It's only big C available, right? There's no other gametes, allele. Small d or small d, that's 1 choice. Big E or small e, that's 2 choices. And big F, small f, also 2 choices. So we multiply all these together. The answer is 16. Does anybody have any question about how I get that answer? It's important that you be able to do this on your own with a different genotype. Any question at all? Okay. Next, we have a cat with long tail breeds with a cat with no tail. All the kittens have short tail. What inheritance pattern best explain this? Now, anytime you see more than one phenotype, right, you should automatically be thinking about codominance or incomplete dominance. So in this case, the middle version, right, if you would, is somewhere between a long tail and a no tail. So whenever you have like an intermediate phenotype, you would call that incomplete dominance. Okay, this is where you, it's like the pink flower situation, right? Yeah, it's somewhere in between the 2 cases. So like, I could call this big L for long tail, small l for no tail, and big L, small l would be for the intermediate, right? The heterozygous phenotype. Okay, next. Brown eyes is dominant over blue eyes.
Sir,
Yeah.
for the incomplete dominance, how would you explain that on the assessment, or do you just say incomplete dominance? That's it.
Oh, yeah. You can say this is incomplete dominance, and if you want an explanation, you can say the heterozygous phenotype is an intermediate expression of the 2 homozygous phenotypes. Okay, it means it's somewhere in between big L and small l. That's what this means. Does that answer your question? Was it Janice?
Yes, thank you.
All right. All right, brown eyes is dominant over blue eyes. Is it possible for two people with brown eyes to have a blue-eyed child? Okay, so if this is dominant, then that means big B is brown eyes and small b is blue eyes, right? So is it possible for two people with brown eyes to have a blue-eyed child? Well, you got to think this through carefully, right? Just because they say brown eyes, it doesn't automatically mean like big B, right? What if they are big B, small b? If one parent is big B, small b, and then the other is also big B, small b, then there's going to be a 25% chance the child will have blue eye, right? So yes, if parents are both big B, small b. And you go ahead and do the Punnett square, you see that it's 25% chance. Okay, let's take a look at this here. Janet has normal vision. Her husband has normal vision. Is it possible for them to have two sons where one has normal vision and then the other one is colorblind? Okay, so if Janet has normal vision, then Janet must have at least one normal copy of X, right? The husband, who has no name in this question, also has normal vision, so he is gonna be XY for sure. Okay, so like we don't know what the other one is, but you know, if you read the question, right, is it possible for them to have two sons where one has normal vision, the other is colorblind? You know, if there is a colorblind son, then Janet has to be XC, right? The other allele has to be XC. So let's try to, you know, do a Punnett square here and see. X and Y for the husband, XXC for Janet. And if we do our Punnett square, right? Let's look at the sun here. Right? These are the suns, right? There is a 50% chance for the sun to have normal vision and a 50% chance for it to be colorblind. Right? So is it possible for them to have two suns where one is normal vision, one is colorblind? Absolutely. Right? And you can just show the Punnett square to support your answer, right? What about daughters? Right? These are the daughters. Is it possible to have two daughters where one is colorblind, the other is not? Right, take a look at here, right? XC is not colorblind. Sorry, XX, I mean to say XX. XX is not colorblind. XXC, that's also not colorblind, right? So this is no, okay, not possible. All daughters will have normal vision. Any question about that? Okay. Consider two traits, flower color and number of petals. The parent generation is purebred. Okay, so we have pink flower with six petals, orange flower with four petals. F1 generation, 100% orange flower with six petals. So I've said this in class a couple times already. If you start with pure breed in the P generation, then everyone in the F1 generation will exhibit the dominant trait. And let me say that one more time. If you start with purebred parents, everyone in F1 generation will have the dominant trait. So just by looking at the F1 generation, we can see that orange is dominant over pink. Okay, and what's the other one? Six petals is dominant over four petals. Okay, if you cross two F1 individuals, you cross two F1 individuals. If there are 2,000 offspring, approximately how many will have orange flowers with 6 petals? Okay, so in class, right? I told you if you have like a 4x4 Punnett square, I mean, you can do it yourself, but you can also just remember, right? 9 out of 16 will have the dominant trait, and then 3 out of 16 will have, dominant trait and then recessive. 3/16 will have recessive and then dominant, and then 1/16 will have recessive. So over here, the question is asking us to find the, oops, sorry, the program just crashed. Give me a second. Let me restart. Hold on a second, huh? There we go. Oh, totally just lost everything. 3/16 will have the dominant trait for the first one and recessive for the other. And then another 3/16 recessive dominant, 1/16 is recessive, right? So in this particular case, we are looking for how many will have orange flowers. Orange is a dominant trait with 6 petal. That is also a dominant trait, right? So dominant, that's 9/16. So we're going to go 9/16 times 2,000. Okay, just going to go ahead and plug that in. And that works out to be 1125. Okay, if they're looking for, say, pink with 6 petals. Okay, pink is a recessive trait. 6 petal is dominant. Recessive dominant, that's 3 out of 16. So you would do 3 out of 16 times 2,000. Okay, if you don't understand what I mean by this, then you can make your own Punnett square and then you can like color it in, right? So you see that's the 9 of them, right? And so on and so forth, right? Like we did in class. Does anybody want to ask anything to clarify for number 9? It's absolutely important that you are familiar with this for the test.
Sorry, how do you get the orange is dominant to pink? Is it redder?
Sorry, how did I get the
I found this on the web for how do you get the orange.
sorry, once again, Sarah, please. I just didn't quite catch your question completely. Maybe one more time, if you could, please.
Okay. You said that orange dominant to pink, yes?
That the orange is dominant to pink. Oh, you asking me how do I know orange is dominant for pink?
Yes.
Okay, so if you start with pure bred, okay, pure breed means they are homozygous everywhere, okay. The F1 generation will always have the dominant trait. Okay, so if you take 2 pure breed parents and you breed them, the F1 generation will always be showing the dominant trait. So because the F1 generation are orange and they have 6 petals, that's how I know orange is dominant over pink and 6 is dominant over 4. Do you understand?
Yes, thank you.
Okay, yeah. All right, now this is another one that is get people all tied up in a knot. Consider the cross this and that. Okay, what's the probability of producing the genotype that? Okay, we did this in class. Actually, it's one of those poll questions, but let's go through it again, right? We have 3 separate genes here, and if we are gonna do the Punnett square, it's gonna be too much, right? It's going to be like 8 by 8, which is 64 squares, right? So instead of doing that, we are just gonna do each gene separately. We'll do the A's, we'll do the B's, and we'll do C all separately. So we focus on the A, then we have big A, small a, cross big A, right? You see that? And then we're gonna go ahead and do the Punnett square. big A, small a, big A, big A. And it's just, you know, same stuff. So which one are we looking for in our result? Well, we're looking for the big A, right? So big A is all here. And that's going to be 1/2, right? 1/2. Now we're going to do the same thing, but for B. So we have big B, small b cross big B, small b. Okay, we'll go ahead and do the Punnett square one more time. And which genotype are we looking for in this case? We are looking for big B, small b, right? So that's going to be 25%, sorry, 50%, 1/2 again. We're going to just complete this with the C. Big C, small c, small c, small c. And we're looking for big C, small c, which once again is going to be 1/2. Okay, so the probability of getting all three of these together is going to be half times half times half, which is 1/8. Okay. Does everybody understand? Okay, so you just do each gene separately. Each gene has its own Punnett square. You find out the fraction, the probability for what you're looking for, and then you just multiply all of them in the end. Okay. All right. Next, hemophilia is X-linked recessive bleeding disorder. Use the following information to create a pedigree. Alice has hemophilia and she marries Terry, who is normal. Okay, so let's draw that. Here is Alice, and she's gonna marry Terry. Okay, so Terry is normal. We know that means he is going to be XY. Alice has hemophilia. Okay, so we say XH, XH. If it confuse you because I'm using a different letter, right? You can stick with XC, XC. It doesn't matter. Okay, I just the subscript just you know, you just use the first letter of the disease, right? So she has hemophilia, which means we're gonna shave her in, right? Like that. Okay, so what's going on? They have 2 daughters and a son. Okay, so we're gonna have to create 3 separate shapes here. We have how many? Sorry, what is it? 2 daughters. Okay, so and 1 son. We're gonna put the son right here. Daughter number 1, daughter number 2. So let's see what's going on with the daughters. One of the daughters married a man, and they have a hemophiliac son and a hemophiliac daughter. Okay, so that's going to be the daughter who's going to be marrying a man, and the man is going to have a son and a daughter. Okay, and both of them has hemophilia. So we're going to have to shade them in. Okay, so that means this son here, grandson, if you would, that would be X H Y, and then this would be X H X H. Any questions so far? about, how I know any of these information. It's not done yet. Okay, we still have to complete the map. Anything? Okay, so now we're gonna work backwards, right? Where did the XHXH come from, right? Okay, so one of them has to be from the mother and the other one has to be from the dad. Okay, and the other allele must be Y. Okay, so actually, even though the question didn't tell us, right, we know that he is going to have hemophilia. And this XH came from Alice. She's the only one who has the hemophilia gene. And the other X would be from Terry. So this daughter for sure is going to be a carrier. Okay? We don't really care about these other kids, okay? Because the question didn't ask us about it. But we know for certain she's also going to be a carrier, right? Because she's going to get the XX from Alice and the normal X from Terry. And then this son, even though we don't have any information about him in the question, we know for sure he is going to be also a hemophiliac, right? Because he's going to be getting the XX from Alice. Okay, so the question is asking, what's the probability that their next son is going to have hemophilia? Okay, so we can go ahead and do the Punnett square to figure it out. Right, so we put the XXH here, XHY. So if you look at just the sons, right, there's a 50% chance the next son will have hemophilia. Okay. I hope that makes sense. Let's take a look at the next one. How many autosomal chromosomes are found in a normal human gametes? Okay. Autosomal chromosomes are chromosome 1 to 22, okay, not including the sex chromosome. So how many of those are going to be in the gamete? 22, right? Sperm or egg, right? How many chromosomes are found in normal human cell? That is in the prophase, 46. Even though you have duplicated the chromosomes, but they're stuck together at the centromere, right? So you still count them as one. How many chromosomes are found in the normal human cell during anaphase? So in anaphase, the sister chromatids have separated and you count them separately. So now there are 92. How many sex chromosomes are found in a normal human zygote? 2. How many sex chromosomes are found in normal human gametes? 1. Either carries in X or Y. How many chromosomes are found in the cells of an individual with trisomy 18? So normally there are 46 chromosomes, but this is trisomy 18. That means there's an extra chromosome 18. So that's going to be 46 plus the extra one, 47. Okay. All right, pay attention to this particular one. We did it in class, but it's good to go through it again. An organism has chromosome numbers of 2n. Yes, Sarah, go ahead.
Get the 47 chromosome on F.
Yeah, sorry, what's the question? about F?
How can you get here 47?
Oh, okay. Trisomy 18 means there are 3 chromosome 18 as opposed to 2 chromosome 18, right?
Yeah,
Okay, so normally we have 46 chromosome, 2 of each, right?
yeah.
But now there's
Yes.
an extra chromosome 18, right? So in total there would be, you know, the 46, the regular ones, and then the extra chromosome 18. So that makes it 47 in total.
Oh, okay.
Yeah. The 18 doesn't matter. Like this person can have like trisomy 20, and then we will the answer will still be 47. Okay.
Okay.
Okay. An organism has chromosome number of 2n equals to 12. Okay. So the 2 here tells you the number of sets. Okay, and this is like total chromosome number. Okay, so is the organism haploid, diploid, or tetraploid? Well, it says 2n, right? 2 sets, right? There's a question. Yeah, go ahead, Temi. I don't know who Temi is. But go ahead. Yeah. So yeah,
Sorry, when you say 22 chromosomes, do you mean 22 pairs? And
sorry.
one,
Which question are
I
you
don't
talking about? Which question are we talking about?
know the last of one,
The first one.
number 12.
Yeah.
Number
How
12.
many
Do you
autosomal
mean,
chromosomes? Is this
yeah,
the one you're
do
asking?
you mean 22? Yeah.
Yeah. So what's the question? Sorry,
Do you
louder,
mean,
please.
do you mean 22 pairs of chromosome?
No, I don't mean 22 pairs. I mean 22 chromosomes because this is gametes, right? In gamete, it's only half, right? It's only half the genetic material. So you're not getting 22 pairs. You're only getting one of those pairs, right? Do you get it?
Okay.
All right. Good question. Okay, so back to this one. Is this is this thing haploid, diploid, tetraploid? What is it? Guys?
Diploid.
This is diploid because it's at 2, right? 2n, right? If it's 3n, then it's triploid. If it's 4n, then it's tetraploid. Tetraploid. So 2n, diploid. How many sets of chromosome does a somatic cell have? Well, if it's diploid, it's going to have 2 sets, right? How many chromosomes does the cell have? Okay, so that's the total number, 12. How many homologous chromosome pair does the organism have? Well, there are 12 chromosomes and you have 2 sets of it. So 12 divided by 2, right? That tells you there are 6 homologous chromosome pair. Okay. How many chromosomes does the gamete of this organism have? Right, you take the number divided by 2, you will get 6 as well. How many chromosomes does a cell undergoing metaphase have? If it's undergoing metaphase, then it's just going to be the same as the original, right? 12. Okay, if it's in anaphase, then it's going to be double because the chromosome has separated. Any question about this? Okay, I'm going to put like anaphase here. The answer is 24, right? Okay, because in metaphase, they're still stuck together like that. Right? But in anaphase, they will have separated. Right? So you count them separately, right? Once they are separated. Yes, Sushma, go ahead. You have a question?
So I didn't understand the C1. How many chromosomes does the cell have? How come it came 12?
Yeah, so like we are 2n equals to 46, right? We have 2 sets of chromosome. Each set has 22, right? 23 from mom, 23
Yes.
from this. So that's how we get the 46, right? 2 times 23. So this number is always the total. Right? So in this case, 2n, 2 sets, and the total set is 12. Right? So 6 from each parent for this
Okay.
organism. Okay.
Okay, yeah, thank you.
All right. Hey, make sure you know this one, guys. All right. Moving on. What else do we have? Oh, that's it. That's it for the genetic stuff. So, like, remember I told you, right? Like you have to solve some genetic problems, right? So please, make sure that you review those. Make sure you are able to create your own Punnett square and stuff like that. Okay. I think most people did it well last on the quiz. You guys lose marks for that pedigree question where I asked you to determine the inheritance pattern, right? That's where most people lose marks for. You guys also lose a lot of marks with that flower question in the end, right? If the florist wants to get the most pink flower, they should start with the red flower and the white flower. Okay, red flower is big R, white flower is small r. If you do the Punnett square like this, right, then it's going to be like 100% pink, right? Okay. Don't worry about being able to tell whether the inheritance pattern is dominant or recessive. That's not on the exam anymore. Okay, it's not going to be on a final test, I mean. Okay. Anyway, I don't know if that's good news for you or not. Let's go on and do the biotech stuff now. Okay. What is the purpose of PCR? How does it work? We start with 50 copies of DNA fragments. How many will we have 10 cycles later? Okay, so the purpose of PCR is to copy DNA basically. Okay, a segment of your choice. Okay, how does it work? I'm not going to ask you to describe how it works, okay? But like you should know that you need to put primers, right? You need to use temperature to separate it, right? Just basic stuff, right? If you start with 50 copies of DNA fragments, how many will you have after 10 cycles? Okay, so 50 copies, okay, multiply by 2, right? You're going to do it for 10 cycles. So that's 2 to the exponent 10. Right? You can bring you will need a calculator for the test. So make sure you bring one, and this is going to work out to be this many copies. Okay, so always have the starting number here, and then you multiply by 2 to the exponent of the number of cycles. Okay, so starting number multiplied by 2 to the exponent number of cycles. Okay. What are restriction enzymes? They are going to basically cut DNA at specific sites with certain sequence. Okay, so each one has a recognition site. They recognize a particular sequence and then they're gonna cut it. Okay, which enzyme listed below creates sticky ends? Equal R1, right? Sticky ends. This one and this one. These are the ones create sticky ends. And then these are the ones that creates the blunt ends. Which one are more useful? I mean, that's actually poorly phrased now that I look at it. I mean, they both have their own application, okay? But for inserting a gene, that's the only one method we use. For inserting a gene into a plasmid, right? Sticky ends are more important. Okay, because then we create the complementary sticky ends. They will be able to bind to each other, right? And after you add the ligase, you will be able to insert the gene into the plasmid. Okay. You absolutely must know how to draw like gel diagrams. Okay. Yeah, like I told you, right? In written response question 4 and 5, you would have choice, right? So question 4, I think I just need to double check so I don't tell you the wrong information. Give me a second, okay? So for question number 4, it is going to be 2 choices, okay? Choice A and choice B, but both questions will be related to the genetic unit, okay? Lecture, I mean, okay? So question 4, you have choice A and choice B. You do one of them, but both of them are related to the genetic stuff, okay? Like the inheritance stuff, I mean, okay? Lecture 8, understand? And then question 5, choice A and choice B will be related to biotechnology, okay? And whether you choose A or B for question number 5, you will have to draw a gel diagram, okay? So make sure that you understand how that works. I'm gonna go through it with you now. Consider the following DNA sample treated with a restriction enzyme. Okay, this and this. All right, so let's see. So this is the treated with ECOR R1. Right. So I'll give you one of these tables if you need to do restriction enzyme questions. So ECOR R1 recognize GAA TTC, and GAA TTC is right here. So that's going to give it 1 cut. GAA, right, TTC like that. So that's going to be 1 cut, and I don't think there is another recognition site anymore. Okay, so you have two fragments. Okay, we have like a short one here, and then we have like a longer one over here, longer one. Okay, I'm only going to shade in the shorter one. So over here, we're going to load the sample. That's what the question says. You're going to take sample number 1, you're going to load it into the first lane, right? So we have a short fragment, and then we have like a longer one. Of course, the longer one's going to be on top. Okay, you don't want to put it like all the way to the top, okay? Because then what if in the next what if in the next lane you have a even longer fragment, then you run out of space, right? So you want to put it like a little bit further down, and then you have one that's like even further down, right? So that's going to be the green fragment here in my diagram. Does anybody have any question about how I came up with this? And let me no, that's just gonna mess it up. Just gonna leave it the way it is. Okay, so we have the short one which corresponds to this end, and then we have the longer one which is gonna be on top, but only 2 lines, right? Because you only cut it once. Are we okay? Yes, Tammy, go ahead. Yeah, see your hand up. Yeah, go ahead.
Can we label it as longer one for this one, or how do we like?
Oh,
show
you don't have to
different-
show me. You don't. I mean, like you don't have to highlight it on the test. Okay, you can just draw like that. Okay, like I will be able to tell one is long, one is short. Okay, so like the highlighting part, it's not mandatory, but I'm just doing it to emphasize for you. Okay, is that what you're asking? Okay, treat the sample with SMA1. So SMA1 is right here, and it's gonna recognize GGG-CCC. And there's only one GGG-CCC right here, and it's gonna cut like that. And I'm gonna get a short piece, and this is shorter than that, right? I mean, you can count the nucleotides, but then, you know, you can visually see that this is shorter than that, right? So in the second lane, you're gonna have one that's further down, okay, and then you're gonna have one that's a little bit higher up, right? Because if the blue one is shorter, then the other one is gonna be longer than the first one as well, right? So there you have it. These are the two bands that you will get for the second lane. Next, if you treat it with ALU1, what is ALU1? ALU1 is going to recognize AGCT here. AGCT. I think there's only one AGCT somewhere here. AGCT, yep, there it is. It's going to cut right there. Okay. So you have two cuts, and they're relatively roughly the same size. Maybe one is a little bit lower than the other. Definitely longer than the blue and the green, but definitely shorter than the other fragment, right? So I'm gonna say it's somewhere here. Okay. And finally, we have treating with equal R1 and SMA1. So equal R1 is gonna cut right here at the beginning, just like before. It's the same sequence. And then SMA1 is going to cut it right here. So when you do that, I will get the same blue fragment as before, and I'll get the same green fragment as before, right? So when I mark your paper, I would need to see that in lane number 4, you would have a band that's exactly where the green one was in lane 1, and exactly a blue band where lane number 2 is. Okay, and then the third one is probably somewhere up here. Okay, negative to positive. Does anybody have any question about this gel thing? Okay. You might want to rewatch that on the video. You know, when you're reviewing, it's going to be at the around the 40-minute mark of the recording, I think. If you need to watch that again. Okay, moving on.
Sir,
Yeah,
is the video that you're talking about this one that you're
yeah,
recording?
this one
or
that we're
some previous-
recording. Yeah, I'm just looking at the
Oh,
clock.
will you
Yeah, I
post
think.
this one after?
Yeah, I will right after. Yeah,
Okay, thank you.
no worries. Around the 40-minute mark. Okay, give an example of a palindromic sequence that's 6 nucleotides long. You know, I won't ask you to create ones on the test, right? But you have to know how to recognize them, right? Okay, so like, you know, TTA, and then you can write it backwards, right? TTA, and then we just fill it in using the complementary. Okay, so that's a palindromic sequence. Okay, so on the I'll tell you what the question is. Doesn't matter. On the test, I'll give you a sequence, just one side, and I'll be like, is this a palindromic sequence? Okay, they're like A, B, C, D, E or something, right? You will have to find the palindromic sequence. If you have a hard time trying to do it in your head, just write out the complementary strand and see for yourself. Okay. You know what? Let's just leave them there. Okay, next. 3 different DNA samples are treated with EcoR1. What is the purpose of the marker lane? This is the marker lane, right? The purpose of the marker lane is to provide a reference to measure DNA size. Right? Like if you get a band here, right? Like how big is that? I wouldn't know unless I have a marker length, right? These are predetermined DNA fragments with predetermined length. So if I compare it, I'm like, oh, it's about 800 base pair. Okay. Which sample has the longest DNA strand? Right? That would be like this one. Okay, because it it's like closest to the well, right? Oh, actually, you know what? I didn't read the question carefully. Which sample has the longest DNA strand before being treated with equal R1? So this one, we're gonna zoom in a little bit here, and we can see, okay, that's like 1,000 base pair, right? So that's 1,000. Over here, that would be 800. This is 200. Right, you see that? 800 and 200. So 800 plus 200, that's also 1,000. And then over here for C, it is 600 plus 300 plus 100, which is also 1,000. So before they are being treated, they are all the same size, actually. Okay, they're all 1,000. Okay, all the same size. Okay, but if I'm just looking for like the longest fragment, like after you cut them, right? This is the longest one, right? Because it's closest to the well. What sample was not cut?
Sir,
Yeah.
sorry. How did you get the how did you know which ones are together?
Oh, because each
Like-
lane, right? Like that's one sample, right? That's
Oh,
a
okay,
sam- yeah.
so
So
how did you
should-
match it to the
How do I what? Sorry.
how did you match it to the base pair?
Oh, to the leather?
original base-
Yeah. So
Yeah.
like, yeah, you just have to go sideways. Don't worry about it. It's, you don't have to do this on the test, but you just go sideways. Right? You
Oh.
just find the bar and look at the number. Okay. All right.
Okay.
What sample was not cut by restriction enzyme? That's this one, right? Sample A. How can you tell? Only one band. Like that, you have to know. Okay, only one band. By looking at a lane, you should know how many times it was cut. Okay, so it wasn't cut. And over here, how many times did you cut it? How many times did we cut this sample?
One.
One time. That's right. One cut. One cut is 2 lines, and that's like 2 cuts, right?
Mhm.
That's it. Explain what will happen if you make the following mistake while doing electrophoresis. Forget to add the loading dye. The loading dye is the blue stuff, right? Then what happens here is a sample won't sink into the well. Okay, you will lose some sample. This is
Excuse
type.
me.
Mhm.
For the previous question, would you repeat one more time? How do you get 1 cut and 2 cuts?
Okay, sure. So this is DNA sample, right? Their DNA sample. Right? If you cut it once, then you have 1 fragment, 2 fragment, right? 1 fragment, 2 fragments. Right here, we'll cut it with a white pen. Okay, look, I cut it once. Now I have two bands, right? 1 and 2. I cut it twice. I have 1, 2, 3, right? So the number of fragments that you get is always number of cuts plus 1. If we're starting with linear DNA. Does that make sense?
Yeah, thank you.
If we have circular DNA, if we have a plasmid, right, then 1 cut will give you 1 band, 2 cuts will give you 2 band, 3 cuts give you 3 band. Okay, so you must be careful. Think about whether you're dealing with linear DNA or circular DNA to begin with. And the question will always tell you. If you reverse the positive and the negative terminal, it will run in the opposite way, and you will lose the sample, right? DNA exits at the top, sample loss. Left the power supply on for too long. Right? This time the DNA will go all the way to the bottom of the gel. Right? Just keep on moving, and then it will all come out at the end. Okay, so DNA exits at the bottom. Sample loss. Forgot to add ethidium bromide. This is the staining agent that makes it fluoresce under UV light, right? So if you forget to add that, this is not too bad. You just can't see can view the DNA under UV. That's all. Any question? Yeah, go ahead.
Sorry, what's the work of the loading die?
What's the function of the loading die? Yeah, the loading die makes the solution heavy. So when you add it at the top, right, to the well, when you add your sample here to the well, it will sink into the well, right? If you don't add the dye, then you add it, it will just like poof and go all over the chamber. Okay. All right, here's another question. There are 2 DNA samples with short tandem repeats, polymorphism STR. Sample A contains CAG repeated 10 times. And then we have sample B containing CAG repeated 15 times. Which sample will be further down the gel when separated using electrophoresis? We did this already in class, actually. So this one will be further down. Why? Because it's shorter. Okay, so this is going to be 30 nucleotide, while this is 45. Okay, shorter it is, the further it will run down the gel. The plasmid below is treated with EcoR1 BAMH. When the digested plasma is separated using gel electrophoresis, how many bands will you see? Okay, so we just talked about this a moment ago. When you cut with EcoR1 and BAMH, that's 2 cuts. 2 cuts means 2 bands. Right? 2 cuts, 2 bands. Next one. A gene is inserted into the plasmid, shown in question 8. The plasmid is transformed into E. coli, and then you culture it in the presence of tetracycline. What do you expect the result to be? So we are inserting using EcoR1. So the gene goes right here, right? Okay, so if you insert using EcoR1 and you grow them in tetracycline, what happens? Right? You will have some growth. Right? Because not every bacteria will accept the plasmid. Okay. Those with the plasmid, however, will gain resistance to tetracycline. Okay. So that's why you will have some growth. Right? Many will die because they did not accept the plasmid. Will the outcome be different if you insert the gene using small 1? The small 1 is right here, right? Yes. Everything will die. Right? No growth. Right? Even if they accepted the plasmid, there will be no growth. Why? Because inserting with SMA1 disrupt the tetracycline resistant gene. So even if they accepted the plasmid, they will not have resistance to tetracycline and therefore they will die in the presence of tetracycline. Does that make sense? Okay. Let me see. That's it. That's it for the biotech stuff. Let's go through the rest of it. I think we should have enough. We should have enough time. Okay, let's do the microscope lab. Here we go. Describe the relationship between object and image, right? You should know that by now. It is inverted, which is upside down and backwards. So what would the H look like when it's under the microscope? Like if you have trouble on the test, just draw it out and then you rotate the paper 180, right? Okay, so like you just draw it out normally first. Okay, like this. And then let me see if I can do this. And then you rotate the paper, your paper. You rotate it like this. Rotate it and then rotate it one more time. That's gonna give you the answer. Okay. Same thing over here. Look like that. Okay, and then the P is gonna look like this. Oh, I don't think the E is right. The E is gonna be hold on. I'm having trouble with this one as well. I think it's gonna look like this. Yeah, that's more like it. Yeah. Do you understand? Okay, total magnification. How do we calculate a total magnification? Total magnification is the ocular lens multiplied by the objective lens. Ocular lens is always 10 times and multiplied by objective. For the microscope we use in class, the objectives comes as 3 separate power, 4, 10, or 40. So if you're using the 10 times objective lens, then total is gonna be 10 times 10, which is 100. Okay. So you should know that. A ruler is viewed under the microscope. Find the diameter of each field. Okay, so let's see. I would be, 1, 2, 3, 4 and a bit here, right? So we'll say maybe like 4.1, 4.2, 00 micrometer. It's 4.2 millimeters, right? But then if you times 1,000, it's gonna give you the micrometer. This was 1, 2, 3, 4, 5. That's 5,000 micrometer. Right? 5 millimeter if you want. That's like 1, 2.5 here. So 2,500 micrometer. All right. The DFOV at 40 times magnification is 25,000. What is the DFOV at 400 magnification? So we're gonna use that formula, right? I will give you the formula on the test. You don't have to memorize it. So DFOV1 times magnification 1 equals to DFOV2 times magnification 2. Okay, so DFOV1 is going to be 40 times, it's going to be 2,500 at 40 times, which equals to DFOV2 times 400 times. So we're gonna take 2500 multiplied by 40 and then divided by 400. And if you plug that into the calculator, that's gonna be the final answer. Okay. Next question. The DFOV is 500 micrometer. That means from here to here is 500 micrometer. Find the length of the cell. So how long is this cell? Right? If you eyeball it, you can see that there are, you can fit two of these cells across the center, right? One cell here, one cell here. So 500 micrometer divided by 2, that means each cell is 250 micrometer. That's all. That's all the microscope question you have to know.
Sir,
Mhm.
do you have to eyeball like this, like how many cells you can fit?
Yeah, you can just eyeball it. Yeah,
Okay,
you don't have to do the ruler thing we did in class. Yeah.
okay, thank you. Just do that.
Okay, that's that. Let's do the cellular reproduction stuff. I'm just gonna skip some of these things because some of this is pretty straightforward, right? What is a correct model for DNA replication? Semi-conservative model. You should know that by now. You don't have to know any of the experiment, right? Look at the exclusion list. Okay? I'm not gonna ask you on those. Name 3 differences in DNA replication between prokaryote and eukaryote. We have a slide on that. So for example, multiple origin of replication. In prokaryotes, in eukaryotes, right? Whereas there is only one in prokaryotes. Because eukaryotes has like a lot more DNA, right? So it makes sense to have multiple origin of replication. There are more enzymes involved in eukaryotes, right? And also, chromosome ends get shorter every time DNA replicates. Again, in eukaryotes. And that's because of the RNA primer, right? Nothing is replacing the RNA primer on the lagging strand, right? We discussed this before. Draw a series of pictures to show the process of mitosis. You don't have to draw it on the test, but I'm gonna give you a series of pictures and you better know which one goes first. Okay, so I'm gonna give you a bunch of pictures and then you're gonna have to reorganize it for me, okay? And tell me the right order of mitosis. So I'll leave you to that. You know, some of the things that you should know the definition for. Right? Homologous chromosome, those are the chromosomes that are the same length, same size, with genes in the same order. You get one from each parent. Alleles, different versions of a gene. Big B, small b, right? Those are different alleles, right? Autosomal chromosomes, those are chromosomes that determines all your trait except for your sex. So chromosome 1 to 22, those are autosomal chromosomes. Sister chromatids is what you get after you duplicate your DNA. When they're stuck together at the centromere, each half is called a sister chromatid. Okay. A lot of things here. Explain the significance of telomeres and telomerase. Okay, so let's do that first. Telomeres, oh boy, look at all these numbers. Telomeres are the basically they're short tandem repeats, right? That are located at the end of chromosomes. They are TTA, GGG, right? And it prevents loss of genes located at chromosome ends. Every time you copy your DNA, the chromosome gets shorter and shorter and shorter. Right? So by having the telomeres at the ends, instead of cutting into important genes that are located at the end of the chromosomes, you're just cutting away the telomeres a little bit at a time. Okay. Yeah, go ahead.
Sorry, do we have to remember the TTA GGG for telomeres?
Yeah, I will remember that. Yeah.
Okay,
TTA,
thank
GGG,
you.
yeah. So what was I talking about? Yes, so like if the telomeres are critically short, then the cell is not allowed to divide anymore, right? Because if you divide one more time, if you copy your DNA one more time, you will start eating into important genes, right? So that's what telomerase is for. Okay? Telomerase is an enzyme that re-lengthen telomeres. Okay. In stem cells and germ cells. Right. So when we produce gametes, the telomeres are reset. During gamete production. So the next generation will have fresh, brand new telomeres, right? How is this related to aging? So like I said, when telomeres are cells with critically short telomeres cannot divide. Okay, they become senescent, right? I said in class they become old basically, and when your body has a lot of these senescent cell, right, that causes us to age. It's part of the reason why we age. How does this relate to cancer? One of the characteristics of cancer is limitless replicative potential, right? So telomerase is expressed in cancer cell, right? Allowing them to divide continuously. Okay. Normally, if you divide too much, the telomeres get short and you won't be able to divide when you reach the threshold. But in this particular case, because they have telomerase, it just keeps on re-lengthening the telomeres. Okay. If you look at the telomeres of cancer cells, they are very long. Okay. How does this relate to Dolly, the clone sheep? So because Dolly was born from the nucleus of a somatic cell, right? So the telomere length of that nucleus from that cell is the length that Dolly cells is going to begin with. So instead of getting fresh telomeres, right, Dolly was born with shortened, not like critically shortened, but nevertheless shortened telomeres. Okay, so for the cell that they took from the mother, right? Maybe the cell has been dividing for, I don't know, like 10 years already. So that's 10 years of telomeres erosion, and that's what Dolly was born with. And as a result, right, it die, you know, it has a lifespan that's half of what a normal sheep would otherwise have. So that's that. Okay, any questions about telomeres, telomerase, or anything? Fantastic. Purpose of mitosis, I mean, you should know that by now, right? Okay, to growth and to repair for asexual reproduction, stuff like that. Okay, review your notes. What is BRCA1? BRCA1 is a tumor suppressor gene. That repairs DNA damage. It's going to repair the DNA. If you cannot repair the DNA, then the cell is supposed to stop dividing, right? Okay. So that's what makes it a tumor suppressor gene. Okay, that is that. Let me see. Okay, next one is the protein synthesis here. So what have we got here? A DNA segment has 300 nucleotides. How many amino acid does this DNA segment encode? What assumption did you make? Well, 3 nucleotide makes 1 codon, right? And 1 codon makes 1 amino acid. So if you have 3 nucleotides, you're gonna have 100 amino acids. What assumption did you make? No introns. Okay, so like if everything here is expressed in the DNA, right, you're not cutting out anything, then you should have at most 100 amino acid. The 24th nucleotide is deleted. How many amino acid will be unaffected by the deletion? If you do a deletion, you're gonna have a frame shift mutation. All of you put that on the quiz, right? I don't know if you are doing it because we did it in the review or you understood what that means. But if you delete something after 24th, right, everything behind, you're gonna have extensive missense. Everything before the 24th, right? Number 1 to number 23, they're gonna be fine. Okay, so that number 1 to 23rd, they are gonna be okay. And if you divide this by 3, you get 7 amino acid plus 2 nucleotides, of course, right? But then that's going to be the 8th amino acid, right? And since you deleted the 24th, that's going to be a different one, right? So 7 of them will be unaffected. A nucleotide is inserted after the 50th nucleotide. How many amino acid will be unaffected by the insertion? Same story here. Okay, so number 1 to 49 is going to be it's going to be okay. And if you take 49 divided by 3, right, 16 of them will be okay.
Sir,
Mm-hmm.
If you insert after the 50th, wouldn't it be 1 to 50 that is unaffected?
Oh, I see what you mean.
Is it-
Yeah, sure. We could put 50, but it's still 16 because then 50 divided by
'cause it's
3 is
not-
still, yeah. Mhm.
Oh,
Yeah, but good
okay.
point. Good point. Good point. Okay. This one, do we have to do this one given the coding strand? No. I think we're okay. I think we did quite a bit of this, and the mutation was done reasonably well on the quiz. You won't have to create mutate, do you? Let me just double check. I don't know if you yeah, you have to know how to do mutation, but I don't think you have to like create mutation per se, but you have to like know how to do mutation questions to answer some of those multiple choice questions. But like I won't give you a sequence and ask you to make a neutral mutation. So that's not going to happen. Okay, but you still have to know how to do it for the for the multiple choice section. There is going to be, of course, a codon chart at the very end. So there's that. Okay, let's take a look at number 3 here. Number 3, hemoglobin is oxygen carrying protein found in red blood cells. Its quaternary structure contains 2 alpha chain and beta chain. Thalassemia is an inherited blood disorder that causes your body to have less hemoglobin than normal. In some forms of thalassemia, the beta chains are completely missing in some hemoglobin. What kind of mutation is likely the cause of this? Okay. When you are completely missing a whole chain, what mutation best explain this?
Deletion.
Gonna need more than that. Because like deletion is gonna mean deletion doesn't mean you delete the whole protein, right? Deletion just means you delete like nucleotides, right? You know what I mean?
Yeah.
Yeah. So what kind of mutation would cause like a whole chunk of protein to just be gone, guys?
Insertion.
Nonsense mutation.
Nonsense mutation, that's right. And you know, the nonsense mutation could be as a result of deletion, could be as a result of insertion, right? It could be as a result of base pair substitution, right? But the key thing to say here is that there has to be a nonsense mutation somewhere. Okay? Remember, nonsense is a stop codon, right? So if you prematurely stop the translation process, you're gonna lose a whole chunk of protein, which actually is what happens in thalassemia. All right, pretty serious stuff. Okay, explain the likely outcome of these things. Okay, so I'm not gonna write everything out here. I'm gonna explain to you, and then you can watch this part again later on if you want to. Okay, so this is like around 1 hour and like 20 minutes if you need to rewatch that later. You might want to write that down, 1 hour 20 minutes on the paper. So the outcome of these things depends on where it happens. Okay, missense mutation means you only change one nucleotide. Okay, well, it depends on where it happens, right? Sorry, not one nucleotide, one amino acid, right? And depends where it happens, it might not have a significant outcome, right? If it happens you know, to a place of a protein that's not vital for its structure, right, then maybe you're not going to notice it. But if it happens in the active site of an enzyme, that could change everything. Nonsense mutation sounds bad, but again, right, if you if this happens towards the end of your sequence, then maybe it's okay. You instead of losing like a half of your protein, you might only lose 1 or 2 amino acids, right? I'm not saying that's going to be okay every time, but you know, it's less detrimental if it happens towards the end compared to happening at the beginning, right? Frame shift, it's usually pretty bad because everything after the frame shift is going to be wrong. You're going to have extensive missense mutation. You might even have a nonsense mutation in your new frame, right? So this one is definitely more devastating. Okay, it will also cause you to potentially miss the original nonsense. To miss the original stop codon, and it keeps on going beyond, and you end up having like all these extra amino acids that, you know, might or might not affect the protein. This one we did multiple times already. We actually review on this as well in unit 3 quiz. Sorry, quiz number 3, I mean, right? So make sure you know what a promoter introns, what all these things are, and you know what is being cut out, adding of the 5' cap and the 3' poly A tail, all that stuff, right? Relationship between the codon, anti-codon, tRNA, amino acid. You know, the codon is the AUG, UGG stuff. That's the one we look up on the codon chart. The anti-codon is the one that is on the tRNA, right? The transfer RNA, right? That will be complementary to the codon, and it's going to be bringing in the correct amino acid, right? Yeah, go ahead, Tammy.
Sorry, what causes the introns on the coding strand? Like,
What
why do
question
they
is that? This question number 5. What about it?
Yeah,
Sorry.
I'm
What?
just asking. what causes the introns? Like, why are they introns on the coding strand? Like,
Oh, the introns is just part of the DNA, right? Like when you copy your DNA, you copy both the coding as well as the non-coding strand segment, okay? And that's why you have to remove the introns before you are able to do the translation, okay? So that's part of the post-transcriptional modification, right? Does that answer your question, or that's not your question? Sorry.
it is.
Yeah. Okay. So that is that. Now, I know I didn't teach you guys lecture 10. I have to remind you, you don't have to know the whole lecture 10, right? You just have to know the anatomical terms as well as the cavities and the planes, right? And there are only 5 multiple choice questions on the whole test for those for that particular lecture. So does anybody have any question about lecture 10 that they want me to clarify, or about anything, you know, for the test before we sign off here?
Sir,
Mm-hmm.
for the worksheet that you gave us,
Mm-hmm.
would it be possible for you to go through number 2? I missed out like from D, E, F, and J. I wasn't too sure about those.
Hold on. You mean for lecture 10?
Yeah.
Okay, hold on. I just gotta pull it out. Give me a second. Which one is it? Number 2 on the second page?
Yeah, the second page, number 2, D to F, and then J.
This one, right?
Yeah.
Okay, well, let's just take up all of it anyways, okay, since we're here. So the cranial cavity is up here. That's where your brain is. And the thoracic cavity is right here, okay? That's where your lungs and heart is supposed to be. Yeah, so the cranial cavity is on top of the thoracic cavity. So we say superior. Superior. The brain is blank to the skull. Skull is like the skeleton, right? The head. And then the brain is inside the skull. So we say is deep. Opposite of deep is superficial. The ears are blank to the eyes. If it's farther out from the middle, we say lateral. The buttocks, the bums, right, are blank to the pelvis. It's on the back, so we say posterior. The legs are blank to the trunk. Right? Trunk is like, you know, your waist, right? Okay, so like the legs are below that, or we say inferior. The heart is between the lungs. Okay, so it's closer to the center. We say medial. Okay, medial is this way, lateral is that way. The elbow is blank to the wrist. If it's closer to the joint, right, like the shoulder in this case, we say proximal. Okay, so like the fingers, which is further away from the joint, we'll say the fingers are distal to the wrist. The calf, which is the, can't really show you, it looks like it's, the camera is not like wide enough, but like the, if you think about like the feet, right? The legs, right? Like the calf is at the back, right? And then the shin is at the front. So we say posterior. Ankles is down here, ankle. Your knee is further up, right? So the ankle will be further from the joint, from the hip joint, than the knee. So we say distal. Your appendix is on the right side of the body, and the gallbladder is on the left side. So they're on opposite side of the body. We say contralateral. Contralateral. If it's on the same side, then we say ipsilateral. Okay, IPSI, ipsilateral. On the test, if you don't know like where the stuff are like in the body, you can ask me. I'll just point to it for you. Okay, if you don't know where the shin is, I can show you on the test. Okay. I mean, it's in terms of difficulty, it's certainly seems more difficult because of all those biotechnology stuff, right? But I expect you guys to do just fine. It will be similar to test one. Okay, and in terms of the format, it's same as everything we've done so far. Okay, so I'll see you all on Thursday, right? 12:30. If you have questions, you can email me. I'll get back to you for sure. Yeah, thank you for coming. Bye for now.
Thank you, sir.
No problem. Have a
Thank
good
you,
one.
bros.
Yeah,
Thank
see
you,
you.
bro.
Thank you. I have
See
a
you
quick
guys.
question.
Yeah, go ahead.
I